Vegetative propagation by understand easily. In the previous video, we discussed on asexual methods like fragmentation, spore formation and budding. In this video, we'll be learning vegetative propagation in plants, which is also a method of asexual reproduction. Let's start. First of all, what do you mean by vegetative propagation? Oh yes, when new plants grow from the vegetative parts of the plant, which are leaves, stems and roots, it is called vegetative propagation. This process is a type of asexual reproduction as it involves only one parent plant. The vegetative part is called propagules. Let's discuss about the vegetative propagation through stems. The stems of some plants grow horizontal to the ground. When nodes start developing on the parallel stem, it starts growing roots and shoots. The roots grow downwards into the soil while the shoots grow vertically upwards. The leaves, roots, buds and flowers start developing. If the stem is growing under the ground instead of horizontally growing, they store food to produce new plants. Examples of some underground stems are tuber, bulb and rhizome. The picture to the left shows the horizontal stem growing parallel to the ground. Once the roots and shoots start developing at the nodes, the other parts of the plant will develop too. Propagation by stem tuber. The underground stems of potatoes, yam and Jerusalem artichoke are called stem tubers. These vegetables have nodes on them called eyes. Each eye bears a bud which grows into a new plant. For example, when potato is cut and planted into soil, each eye grows into a new plant. Propagation by bulb. Stems of plants like onions, tulips, daffodils and lily are called bulbs. Why is it called bulbs? It is because they store nutrient reserves to ensure the plant's survival. These bulbs have both terminal and axillary buds which are protected by fleshy scaly leaves. These buds give rise to new individuals. Propagation by rhizome. Underground stems of plants like turmeric and ginger have internodes, nodes, thin scaly leaves and axillary buds. These axillary buds on the underground stems give rise to new plants. Some other examples of rhizomes are asparagus, ginger, cannas and hops. Some rhizomes are also used in cooking like lotus, turmeric and fingerroot. Vegetative propagation by leaves. Plants of bryophyllum, peperomia, apicia and begonia have buds on the edges of leaves. When these buds detach from the plant and fall into the moist soil, they grow into new plants. By roots. Some plants like sweet potato, dahlia, asparagus, ivy and tamarind have large and swollen roots. These large roots have buds on them which when detached from the parent plant gives rise to new plants. All these methods were natural. There are also some artificial methods of propagation. They are cutting, layering, grafting and micropropagation. Let's begin with cutting. In the process of cutting, the stem having an axillary bud is cut. This process is called stem cutting. Then, the cut stem is planted into moist soil. After a few days, shoots and roots develop from the cut stem. Rose, bougainvillea, chrysanthemum, grapes, sugarcane, bananas and cactus are commonly propagated by cutting. The next method is layering. In the process of layering, the bark or the stem nearer to the ground is bent. This bent part is then covered with moist soil. After a few days, the shoot and root develop on the buried portion of the stem. As this stem can grow independently, it is cut from the parent plant and grows as an independent plant. China rose, guava, lemon and jasmine propagate through layering. Air layering is a method of propagating new trees and shrubs from stems still attached to the parent plant. The stem is wrapped with damp moss to encourage roots to form. Grafting. In the process of grafting, the stem of one plant without the root is cut obliquely. This shoot part is called the scion. Then, the scion is placed on top of the rooted stem of another plant. This rooted stem is called stalk. They both should be placed in such a way that their cambiums are in contact with each other. They are held together strongly by a piece of cloth and is covered with a polythene sheet to avoid infections, diseases and water loss from the cut parts. The advantage of grafting is that it is possible to change the cultivars and get a new and more desirable plant. This method also reduces the hassle of replanting. Last but not the least, micropropagation. Micropropagation is done by the tissue culture method. In this method, a tiny piece of bud or shoot or any other part of the plant is taken. This part is called the explant. It is then treated with chemicals to prevent any viruses, germs or bacteria. Then it is grown in a particular nutrient medium so that the cells of the tissue divide and grow into a mass of indifferentiated cells called the callus. After a few days, the callus develops into the shoot and root. It forms a small plant called a plantlet. The plantlet is then planted into the soil. Plants like asparagus, orchids, cauliflower and chrysanthemum propagate through tissue culture technique. The advantages of tissue culture technique are, it produces superior quality of plants. The new plantlet can grow in a small amount of time. The new plant is more likely to be virus and germ-free. It is not required to be done in any particular seasons and so, it can be done throughout the year. It is useful to grow seedless plants. It can be applied to interspecific hybrid, a hybrid formed by breeding two different species. Here are some advantages and disadvantages of vegetative propagation. The advantages are that it is a quick and easy method. Plants need to less time to mature and bear fruit much faster than those grown from seeds. New plants are exact copies of the parent plant because it includes only one parent plant, asexual reproduction. No fusion of the male and female takes place. Thus, all the good and desired qualities of the parent plant are present in the new plants. Useful for growing seedless plants like sugarcane, banana, pineapple and grapes. Here are the disadvantages. Some APMEs diseases can get transferred from the parent plant to the new plant. Propagules cannot be stores like seeds. Results in overcrowding of plants due to productions of a large number of plants. There is also a competition for sunlight, water and nutrients which affects the growth of the plant. New plants cannot adapt to the changing environment. The picture to the left shows that the process of layering is a fast and effective process.
Previous video on asexual reproduction. To understand this video on Understand Easily channel, link in the description.